Okay, so this is a topic on organelles as part of cells as the basis of life unit. Now, we've already looked at cells a little bit at the start of the course, just so you had a basic understanding before doing DNA and proteins. But now we're going to go back and look in them in a lot more detail. So basically that is the way the course is structured, is that we need to connect all the different parts of it. So this connects to DNA and proteins, understanding where those processes that we've been discussing work and how enzymes contribute to cell metabolism, cell energy, and the whole processes of life that happen inside cells. As part of that, though, you need to have a pretty good background and understanding of all the different organelles and parts of the cell. So in student learning outcomes, we're focusing here on eukaryotic cells and some of the structures within them. Now remember, eukaryotic cells have membrane-bound organelles, and most of what we're talking about here is membrane-bound organelles, although obviously ribosomes, cytoskeleton don't come into that category, but most of the other ones here do. Okay, so, and again, just a reminder at the top there, eukaryotic cells, what we're talking about here is fungi, animal and plant cells. But specifically we're going to focus on perhaps some of the different structures in plants and animal cells. So, often a starting point, we learn about this since year 8, is the nucleus, a really important part of the cell. So the nucleus is a membrane-bound organelle within the cell. Each of these, we have a diagram on the left and an electron micrograph on the right. So this is a transmission electron micrograph used from electron microscopy, basically showing those structures in a two-dimensional form. So this is what they actually look like, and this is how we tend to represent them. So within the nucleus, of course, we have chromosomes for a human. We have the 23 pairs of chromosomes that are unwound as chromatin, so they don't look like X's, they don't even look like the condensed lines. They are free, unbound, wiggly, messed up, tangled chromatin like this. But there is a sort of dark center or spot within that, which is the nucleolus, and this is the site of RNA production in particular. That's where ribosomes are made. Just one more thing. There is, of course, within the nucleus not just DNA, but free nucleotides for making DNA and making RNA, enzymes and all sorts of things in there. Mitochondrion, a very key organelle. Its main purpose is to produce cellular energy in the form of ATP by converting fuels to ATP, basically transferring that chemical energy from one form in glucose, for example, in the bonds of glucose, into a more readily accessible form of chemical energy in ATP. And so this links to endosymbiosis, because the mitochondrion is thought to be a prokaryotic, symbiotic element within the eukaryotic cell that's no longer obviously a free-living prokaryote, but evolved from it. And that's why we have these two membranes, the outer one being from the host, the inner one being from the original invading or symbiotic prokaryote, and also therefore having its own DNA and making a number of different proteins as well. But a lot of the proteins that work in the mitochondrion come from the nucleus as well, so it comes from both. That's sometimes quite a fat one, or sometimes long, thin ones, so they do come in different shapes within different types of cell. And just a reminder that there are many tens or hundreds of mitochondria within cells. So, and they divide by binary fission on their own when the cell is growing. Important part to remember as well is that part of the process of aerobic respiration happens in the cytoplasm, and then the mitochondria takes over and extracts even more ATP from those fuels. In plants, we also have chloroplasts, again connected to the embryo-symbiotic theory. These evolved from prokaryotes that are now living inside the eukaryotic cell, and again, therefore they have their own DNA and double membranes. The process of photosynthesis happens across the thylakoids that are shown here, and so they're very easy to distinguish from mitochondria in electron micrographs because of those thylakoids. You might also see a starch granule. So the reason that starch is there is that the organelle is responsible for making food energy in the form of glucose from water and carbon dioxide. Once that glucose is made, glucose monomers are put together into a polymer, and so starch is a polymer of glucose that plants use to store that fuel that they've made, and then they release the glucose to mitochondria for producing energy. So, strictly speaking, and I know a lot of people get this wrong, the chloroplast does not make energy in the form of ATP. In fact, it uses a little bit of energy to make it. So the chloroplast makes high energy molecules, the fuel, the food, the glucose for the cell, and then transfers that to the mitochondria for making cell energy. So chloroplasts make glucose from carbon dioxide and water using sunlight as an input of energy. While we're talking about plants, the vacuole is a really important one in plants. So it's quite dominant within this diagram or within this electron micrograph. It's a very large organelle. So sometimes it can be confused with the nucleus of similar size over here, but clearly that nucleolus makes a bit of a difference. Because it's filled with water, it comes up quite white on electron micrographs. So it's used to store a whole lot of water, sometimes wastes and salts, and regulates the water balance inside plant cells, which is why they have a large one. Animal and other cells do have sometimes a small vacuole, but the large central vacuole is what we find in plants. And when this is full, the cell is turgid and rigid. When the plant starts to run out of water, it uses up the water in the vacuole, that gets smaller, and the plant starts to wilt. And so by regulating the size of this, plants can regulate their structure. Endoplasmic reticulum is found just outside the nucleus and can be categorized as smooth ER or rough ER, rough ER having ribosomes embedded in it. So the smooth ER is where membranes are made. Obviously there's membranes in the nucleus, in the ER itself, in the organelles, and in the plasma membrane. And that's produced, synthesized, and sent around the cell from the smooth ER. There are also membrane-bound proteins that need to be made, or proteins that also go out in membranes and get exported from the cell. These proteins are made in the rough ER. So if it's embedded in a membrane or it needs to be exported from the cell, that protein is made by ribosomes that are on the ER. And we'll have a look at the whole process of protein production and export because it goes through a lot of different organelles and puts a lot of these ideas together. Part of that journey is going through the Golgi Body. So Golgi Body vesicles come in, a transport vesicle, which we're going to talk about in a minute, will come into the cis phase, go through a number of different stages and then come out. Now those might be being secreted outside the cell for those exported proteins. They might be being sent to the mitochondria or chloroplast if that protein needs to go to one of those organelles. The Golgi Body there is to sort and direct the vesicles to where they need to go. So what is a vesicle? Well, it's basically just you can think of it almost like a tiny cell. It's a lipid bilayer, just like a cell, with a small amount of contents in it. And so if the cell needs to send any materials around the place, this is a little package that it can send around the place. This diagram doesn't show any proteins embedded in there, but you can see this vesicle that was formed by endocytosis, being pulled in from the plasma membrane, has lots of dark stuff around the inside and the outside. That's a whole lot of proteins attached to it. So even though it's a lipid bilayer, just like the plasma membrane, it's embedded with proteins as well. So proteins can be transported by being embedded in the membrane or inside this internal fluid space. So a lysosome is a special type of vesicle that actually has a whole lot of enzymes in it that are going to digest things. So a lysosome is basically a little packet of enzymes that will digest things, either things that have been taken up by endocytosis, like the process up here, and taken inside the cell, or in this case you see a lysosome fusing with a mitochondria. If this mitochondria is no longer operating anymore, we need to digest that and reclaim those amino acids and use them to make functional proteins. So a lysosome will go in, fuse, those enzymes can digest everything that's there and recycle that. So used for a whole lot of recycling of things within the cell. Last one here, not a membrane-bound organelle, the cytoskeleton is a very important structure within the cell. So we can see here these somewhat rigid, somewhat flexible rods and structures throughout the cell. Actually come in three different types. Actin makes microfilaments. There are also intermediate filaments, and then microtubules are the thickest. And they all have slightly different roles. Even though it's called a skeleton, it's not just like a skeleton. So we think of our skeleton as being a rigid, fixed structure that supports the cell, supports our bodies. The cytoskeleton does support the structure of the cell, but it's actually quite dynamic. It grows and shrinks and gets changed all the time as these subunits get added and taken away. So it's pushing and pulling and creating the cell shape. So these fine projections here from cytoskeleton, but also if things are moving around the cell, you need to move vesicles, as we showed before, or organelles around. They'll travel along these as if they're little highways and routes. So it's like a skeleton, but it's also like muscles, and it's also like a transport network. If you go on to the online version of these notes, these images are going to be animated. Unfortunately, it doesn't work in the desktop version, but here is an actin filament being made by polymerization. So you've got lots of these subunits that stick together, as we see up here, to form and remove as that goes along. And here is a microtubule with a vesicle attached to it via this motor protein. And in the animation you see the motor protein walking along that as it pulls the vesicle along. So these vesicles don't just float around in the cytoplasm, they travel along microtubules to go to their destinations. And we can see here with this vesicle it's protein embedded and also has some carbohydrates. Those things help to direct where it needs to go. We've got some additional resources here in Chapter 8. There's a couple of click view videos. In fact, this Cells and Organelles one I want to show in class because it's really useful. There's Mr. Lecornu's organelle video, and this crash course video with Hank, who, I love his presentation style. It's quite dense and rich with a whole lot of stuff. Probably most of it goes, or some of it goes beyond what you need to know, but if you want to know more and get a really solid background in organelles, I suggest watching that one.